In this module, we will start to talk about the second chapter of fermentation. As we know that the fermentation is a process of mass culturing of the organism for the sake of any desired product. So, when we say that mass culturing, so for the growth of the organism that needs some nutrients and other some physical environment. So actually in this introduction of fermentation medium, we will talk about different aspects about the medium. So in this slide you can see that in a process of fermentation, the organism is very important. The main actor of the fermentation process, that is organism, because that has to the mass culture. And then is a medium. In this module, we will talk about in detail about the media. Media is a plural word of medium. And then ultimately, with the association of the medium and then that provide the platform to grow our selected organism, then that will be a suitable process. Through that process which we called as fermentation process, that basically help to facilitate the product formation that we need to the fermentation process on an industrial scale, either a lab scale. So there is somehow difference between a lab scale and industrial scale, but the main theme is the same. So what is the medium? So medium actually the intervening substances through which the metabolic requirement are conveyed or physical forces are transmitted. Actually all these metabolic requirement and the physical forces are transmitted to the organism of our choice. So what does the medium need to do? Actually, as we know that the fermentation process is the mass culturing. That means that in which the organism grow in a mass level. So the grow media help to grow the organism or a microorganism, so it produce the biomass and the product and should not interfere with downstreaming process. When we see in detail investigation, the most suitable medium for the individual fermentations, organisms have some basic needs. Almost all kind of the microorganisms have their needs which we can call as the common needs. First one is the water. Most of the fermentation process, most of the organism required the water. Then all the organism, when they have to perform different functions, then they require the energy. So what will be the source of the energy? And third is the carbon, and then the nitrogen, mineral elements, and possibly some vitamins. And somehow in some cases when the organism is aerobic, then oxygen is needed. In another case, when some archaea and those organisms which require another gases, they need such as carbon dioxide, hydrogen, and then the mixture of carbon monoxide and nitrogen, etc., depending upon the nature of that organism. So water, energy sources, carbon, nitrogen, mineral elements, vitamins, in some cases oxygen, and in other cases carbon dioxide, or we can say that any other gases that need to that organisms. So basically the medium provides us all possible requirement. How we know that what are the basic requirement of any organism which we have to culture in the medium? We know that when we have an elemental composition of all the organism, commonly we know that the major chemical analysis of any organic material are actually the cell. We know that most 70% of the cell is water, and then the major part of the cell is carbohydrate, then there is a nitrogen, nitrogenous compounds, and then DNA and RNA, a nucleic acid material, and some minerals. That is the part of the chemical composition of any cell. So if we look in the chemical composition of carbohydrate, protein molecules and all that, and we know that carbon, hydrogen, oxygen, phosphorus, potassium, and these are different elements that is required by that organism. So these are the elements, these are the nutrients that is required by any organism. So we can say that these nutrients, these elements, some are required in enough quantity and some are required in a trace quantity. So we further divide these requirement into macro requirement and a micro requirement. So as we say that while designing the medium, sometimes the medium have a more water and some media has a less water. When we talk about the types of fermentation on the base of the water content in the medium, we know that we divided the fermentation process into solid state fermentation, submerged fermentation, and static culture or a surface culture. We know that in submerged fermentation there is n quantity of water, and then when there is a surface culture, then almost the media look like the jelly. But in first case, when we talk about solid state fermentation, we know that it's a less moisture. That's about 1:1 substrate to water ratio or less than that. Suppose there is a 10 gram of the substrate and 10 ml is the water. So if water can be have a less than 1:1. So in case when the water content and the medium is changed, then that also directly affect on the fermenter design. So if we are working on a solid state fermentation, we have to use Koji fermenters. On the other hand, if we are using submerged fermentation process having enough quantity of water, then we have to use stirred fermenters and some other type of fermenters which facilitates the submerged fermentation. In third case, when we called as the surface cultures that mostly use in citric acid fermentation, then we can utilize tray methods and others that has the possible ability. So the media is very critical that provide the suitable environment as well as the chemical need of that organism which we have to grow in a fermentation process.